So the second presentation for this session is on the project called Cell-Based Regeneration of Heart. And then the project coordinator is Professor Ronald Li. And the team initiated a Hong Kong stem cell initiative and then aims to focus strengthening the stem cell and regenerative medicine in Hong Kong.
So thank you, Professor Lo, for the introduction. So first, on behalf of the team, I would like to sincerely thank the Research Grant Council for supporting this project. It really had allowed us to jumpstart a lot of scientific and as well as other progresses. So Professor Yao, Professor Chan, and I are going to be presenting to you a summary of our collective scientific and other progresses. Professor Chan, Godfrey Chan, might have some clinical duties and he may or may not be able to make it. And if I don't see him when the time comes for him to present, I'll do the presentation for him. So, and also as a routine disclosure, myself and several other investigators, collaborators, as well as graduated trainees, they hold equity and or get compensated by this spin-out company called NovoHeart. And all appointments and arrangements have been approved by the corresponding universities and reported in compliance to the Toronto Stock Exchange as assessed by independent auditors and legal counsels. So the company was founded in 2015 and it went public in October this year, now with locations in Hong Kong, California, and British Columbia. With that said, I also want to congratulate Dennis Lowe and Alan on their translational and commercial successes. So one difference between our group and all the other funded projects is that unlike all others, we simply, the group did not exist previously in Hong Kong. Meaning that we all had to start completely from scratch, and some of the investigators hardly knew each other other than on paper, and some did not even know each other. And the majority, more than 90% of the trainees, as they were recruited into the program, they did not even know what human embryonic stem cells were. So I was recruited by Sum Li to Hong Kong, and upon landing, two great friends and mentors, Sum Li and Patrick Tam, independently said to me the following: "This is not a popularity contest." And the following two quotes reflect very well the motto of our group. So do what you feel in your heart is right, for you will be criticized anyway. You will be damned if you do and damned if you don't. And for us, there's only the trying, so the rest is not really our business. So this is the motto of our group before and also for going forward. So we don't have to remind any of the audience here that as published on the website, the objective of this theme-based research scheme is to focus academic research efforts on themes of strategic importance to the long-term development of Hong Kong. So I haven't been able to find anywhere on the website about the specific definitions of strategic importance and long-term development. So as such, our group took the liberty of interpreting these terms in our own ways. So just a little bit of history. So in 2010, 2009 perhaps, so we as a group submitted one of the 400 or so white papers to the Research Grant Council to propose the topic of stem cell regenerative medicine. And we formed a delegation and luckily it got selected as one of the topics for further debates, and we formed this delegation of a number of friends and collaborators to defend the topic. And seconds before we went on the stage, someone came to us, literally seconds before we went on the stage, someone came to us and said, "You're not going to get it." I'm like, "What? You're not going to get it?" And for us, we felt like that maybe yes, maybe not, but it's not really going to change what we are about to do on stage. So we went on, and the group basically has been working together ever since, and they continue Roger Hajar turns out to, we've been publishing together over the years, and he's now serving as the chair for the scientific advisory of this spin-out company. And Han is in the audience somewhere. Yeah. So the new KI Center and CityU are now working together to try to promote the platform for biomedical engineering together, and we're starting to have cross appointees at the faculty level. And King has been a good friend as well, although we have never really been working together scientifically, but the groups together have been trying to promote different programmatic initiatives in Hong Kong, helping each other. And Godfrey and the group have new students, new academic as well as commercial projects that are in the play. And Ken Chin, he moved from Harvard to KI, and also we have been working very closely together. And Joe Wu from Stanford, he was in Hong Kong just 2 weeks ago, and he's coming to Hong Kong again in about 6 days or so to explore new ways of working together. So we felt extremely fortunate that the topic got successfully defended, although after we went down the stage, another person came to us, "You're not going to get it." And we're like, "What?" And that person said, "Because you and the team have just upset the committee." I was like, "Upset? What do you mean?" So we were just trying to defend our topic. We were just reasoning. We were arguing. We weren't meant to be upsetting anybody. So that proved obviously to be false because the topic got selected. And also the team very fortunate to be receiving among the first TRS grant together with all the other groups. So this grant, the resources really allowed us to pursue solid relationship with others, including Karolinska Institute at Edinburgh, in addition to existing American ones as well. I'm not going to go over all these. These are given on the poster outside, one of the posters. But we launched projects together internationally, co-organized joint international and local symposia, public lectures, knowledge exchange meetings for high school as well as undergrad and graduate students, exhibitions that involve over 200 students and parents, etc. So one obvious outcome is that, not so reason, but because of this very project, it really facilitated Karolinska Institute to make that decision of coming to Hong Kong first time in their over 200 years of history to set up the so-called Ming Wai Lau Center for Reparative Medicine. And this slide summarizes to you some of the key statistics. So altogether, the group published over 100 papers with a number of them being reviewed as I speak. And we made about altogether 34 international presentations at conferences such as American Heart Association, International Society for Stem Cell Research, winning a number of scholarships and studentships by our students in Hong Kong. And Andy Wong may be in the audience, and he was really a star, and he's about to defend his thesis, finishing his PhD in just a few weeks, and he got several awards by presenting. So we trained altogether 6 MPhil students and 21 PhD students, and about 11 postdoctoral fellows graduated without including 7 of them who got promoted to the faculty, with 3 assistant professors at least that produced, and 1 turned out to be at NYU as assistant professor. And 9 of them went into the industry in China, in Hong Kong, Novo Hart, and also in the US as well. So altogether, because of this project, it led to at least 30 million Hong Kong dollars of new extramural funding, and also a number of other pending grant applications as well. Academic spin-out that I was describing to you, and as such, has created over 30 jobs in Hong Kong. And as I said, not the only reason, but one of the reasons that it helped to facilitate the establishment of the KI Center here, and Cardiac Initiative turns out to be among the first initiative to get launched. So now let me switch gear and tell you more about the science that the project has been trying to do. So this is the traditional paradigm, and we know that the heart is a 4-chambered organ, with the left ventricle being the major one, so heart attack, heart failure are primarily left ventricular diseases. So with that, we are basically just focusing on the ventricle. And if you look at this heart, you drill the hole in the left ventricle, cut out a chunk of reticular chunk of tissue. You take a closer look, you can see that it's a very thick layer consisting of bundles of fibers that can be further digested down into single cells for studying. That was how I used to study the heart as a graduate student. And the approach that we take in this project is the opposite. So we make new human heart cells, followed by employing a range of tissue engineering strategies to try to put the cells back together as tissues and even heart chambers. And with the so-called miniature left ventricle miniature heart, this is what we call the human heart in a jar technology. So I'm not going to bore you with the details. And once again, this is a slide. This is shown in one of the posters outside. But basically, we started by asking showing you 8 major questions in the field at the time when we put together our application. So the project is about learning more about these. So we have made good progresses, or we've obtained better understanding at least for 7 of the questions that we asked, and 3 others turns out to be very difficult to crack, not only unique to us, but in general by the field, and I'll tell you a little more later. So this is the design of our project. It's a business slide, but I'm going to quickly go over it with you. So first we start by getting, let's say, blood samples from patients, and then we reprogram them into stem cell, iPS cell, as HF has nicely presented. And from here on, it's basically everything that we've been doing in this project is that we managed to come up with a way to instruct the stem cell and specify them into human ventricular muscle cells, heart cells, with a very high yield that we were not able to obtain before. And with the cells, we can then use them to study metabolism, electrical as well as mechanical properties, and calcium as the key secondary messenger linking these processes together. And with the single heart cells, we came up with a range of engineering approaches to put them together as 2-dimensional as well as 3-dimensional tissues, muscle strips, patches, as well as chambers, or so-called miniature heart, for disease modeling, for drug screening, and perhaps in future for transplantations as well. So this is the basic design of our 5-year project, and I'm going to be quickly presenting to you the first one, the project 1, which has to do with tissue engineering, and this is basically reiterating some of what I said before. We get blood samples from human patients, we reprogram, we came up with a way to instruct the stem cell reprogram from this blood, as published in 2 manuscripts, to get human heart cells with a very high purities and yield, and within days we can literally generate billions of cells as the fundamental building blocks. And with these single cells, we can assemble them as patches for studying electrophysiology or the electrical properties, and tissue muscle strips, which come in 2 sizes, the small ones and also the big ones that are more physiological, and as published in 4 papers. And this is what you're looking at, the side view that physically contract in front of you. Or we're most excited about the ultimate weapon, so to speak, is to put them together as miniature heart about the size of your thumb that is fully capable of ejecting blood or fluid, and as such, we can make a lot of clinically relevant parameters. So we can make cells, patches for electrophysiology, tissue strip for looking at the force or contractile dysfunction, or a mini heart that you can literally see that as a pump, and you can measure ECG-like signals and measure the pressure. So what are we using them for? What are the applications? The idea is that what you may not know is that the heart is also the major problem why we are not getting newer, more effective drugs, new drugs, quickly enough. Because whenever, it doesn't matter whether it's cardiac drugs or not, even for long cardiac drugs, for chemotherapy, for treating GI diseases, inflammatory diseases, painkiller, arthritis, allergy. So whenever a new drug fails, it often has to do with side effects of the human hearts. And we all know that drug discovery is a very broken process, because whenever a pharmaceutical company wants to develop a new drug, it typically takes about 10 to 12 years To complete the process, you go through clinical trials, phase 1, phase 2, phase 3, investing altogether 2 to 3 billion US dollars, but at the end, more than 90% of the time, it fails very miserably. And as I said in the previous slide, when it fails, often it's because of side effects to the human heart. And why is it the case? We know that animals are animals, but companies and scientists have been depending on animals to tell them what drug candidates work in humans. People can tell you that their genomes are remarkably similar to those of humans, more than 90%, but statistically, we know that when it comes to pharmacological responses, the correlation is less than 5%. So we shouldn't be depending on animals to tell us what drugs work in humans or not. So if human heart is the problem, animals are the problem, we are now proposing, why don't we try to take advantage of the range of engineered human heart tissues that we have designed and to replace supplement animals so that we can reduce the time, reduce the cost, reduce patient harm, and importantly, improve the success rate of identifying the right kind of candidates for humans in the spirit of precision medicine that HF has also nicely presented as well. So this is the foundational science for a spin-out company that several of us investigators of this grant established in 2015. And we were extremely thrilled that the company has been highlighted by, let's say, BBC World as a Hong Kong-based biotech company. And it also received attention from other media presses like Bloomberg, CNBC, BNN, etc. So with that, I'm going to let Professor Yao to take over from here and present to you the progresses for Project 2. And again, if Godfrey is not going to be able to make it in time, then I will finish the presentation.
So as Ron probably already mentioned that compared to the adult human cardiomyocyte, the human ESC-derived cardiomyocyte, they are rather immature. They are immature in many different ways. One of them is, their cell size is significantly smaller, whereas the adult cardiomyocyte is a lot bigger. So over here, one of the things we are doing is we have 2 goals. One is try to, using a number of techniques, try to facilitate the maturations. And another one is we try to look into the mechanism, underlying mechanism, and why they are immature, and we can find out the things. Okay, so the one aim, which is actually done by the PIs in the CDU and Su Han sitting there. And if you have any question, actually, you can ask them. Although this is a biomedical engineering approach, and they use optical tweezer and try to put 2 cells together, and they also use a laser induced fusion of the cells, and so that the cell size, I just mentioned the immature one, is their size is small. You try to make them bigger. So this is a laser induced 2 cell, and it became 1 during the process. The other part, this is the calcium signaling part, was performed by a group in Hong Kong U, Ronald Lee's group, as well as my group in the CHK, as well as Andy Miller's group in the Hong Kong USD. So here we ask a question, and what is immature in terms of the calcium handling, and we found out, and this is the mature cardiomyocyte, this is the ESC-derived cardiomyocyte. You can see that in the adult cardiomyocyte, there is a T-tube, and this is very important for EC coupling, muscle contractions. Whereas in immature cardiomyocyte, the EC, this T-tube is pretty much not existent. So EC coupling efficiency is very low. In additional, and we found out over here, this is sodium-calcium exchanger. This one is much, much higher over here in the immature cardiomyocyte. And on the other hand, a few protein was actually missing. This is an important one, phospholamban, and this is trident, and this is junctin. They are controlling the calcium release from their sarcoplasmic reticulum. This is not really exist in the immature cardiomyocyte. And as a consequence, actually cytosolic calcium concentration in the immature cardiomyocyte over here. They are relatively higher. That will affect the nuclear calcium signaling. So that will affect the translation. That cause them immature. So in addition, we noticed that we all understand that if you're putting a cell in a randomly put into a culture dish, they are going to be randomly oriented. Whereas the mature cardiomyocyte inside of cell, the cardiomyocyte, they are organized in a very nice way. They are aligned basically so that you have a meaningful contractions. And so what we did is we use a mechanical cyclic stretch, kind of make them aligned so that provide a better platform for the JAK screen because there's more mimicking the cardiomyocyte in vivo. And in addition, we found out the mechanism why they can align in certain directions because we found out the important role of the TRPV4 in this process. Now, in addition, and we established 2 disease model with human ESC-derived cardiomyocyte. One of the disease model we established is a cardiohypertrophy model. We all understand that cardiohypertrophy is one of the big risk factors for the heart failure and human death. And we not only established this model, but also we used this model to answer some important scientific questions. It is known that nitric oxide donor as well as sildenafil, which is active ingredient for the Viagra, which can be used in the clinical to treat cardiac hypertrophy. So the underlying mechanism actually is not quite understood. And from this study, actually, we identified one of the very important protein is a calcium permeable protein called ORI1, and we found out that these 2 agents actually act through the ORI1. So ORI1 is the channel leads to the calcium influx and it leads to the hypertrophy. If you can, these agents actually inhibit ORI1, then inhibit hypertrophies. Okay. Now, this is another different disease model. This is a polycystic kidney disease, and this is one of the very common disease. Actually, in among 1,000 people, you're going to have 2 people have such kind of disease. And we used a human ESC-derived cardiomyocyte to establish the disease model, cardiomyopathy model in this disease patient. And this is a normal kidney. Usually this patient has kidney problem, but not only that, they also has 90% of patient has a cardiomyopathy, okay? And we established model, and furthermore, we answered in a very important scientific question, what was wrong in this cardiomyocyte? We found an important protein called PKD2, which is another name of the PKD2 is a TRPV2. This is a calcium permeable channels. They're controlling, they are regulating the ranitidine, the calcium release from the ranitidine receptor. So these were affecting the autophagy. The autophagy is protecting, okay? So if in the disease patient, this protein is reduced or not, mutated, so that you do not have any protection, so eventually it leads to disease. Oh, it shifted.
So, thank you, Professor Yao. So since Godfrey is not here, I'm going to take the liberty of presenting to you the progress for Project 3, which has to do with technologies to improve the therapeutic potential of human ESL, iPSL-derived cardiomyocyte. So Project 3 is really meant for studying the immunology and other therapeutic aspects. So as it turns out, the immunology has been extremely difficult to crack. So this problem really is not unique to our own group, but also a general limitation of the field over the past several years. So really few progresses have been made worldwide because of the compatibility of different animal species and their differences to humans. So in the process In the process, however, we made new discoveries that we did not anticipate. One was that as we employed the so-called kidney capsule model as a standard way to study how different immune cell populations respond to human heart cell transplantation, we accidentally found a way to make kidney beating like a human heart. And I'll tell you more about the surprise that can be used in future in the next slide. In addition, our investigators also found new molecular pathways that underlie development and maturation and also markers for cell purification. So this is the last project. I'm going to be very brief. Once again, it's demonstrated in one of the posters outside. We came up with a way to convert human ESL-derived ventricular cells into pacemaker cells, and we tested in a large animal model as a possible supplement to electronic pacemakers. This is an area that several of us have been working on in the past 15 years or so, initially in collaboration with HF who's in the audience. And we found that by injecting ESL-derived terminally differentiated cells into the kidney, the cells had a hard time surviving. But if we injected progenitor into the kidney, all of a sudden we were able to populate the entire kidney with over 1 billion cardiomyocytes, making the entire kidney beating like a heart sitting, somewhere here in the abdomen of these animals. So now this is a collaborative project with Ken Chen after he has moved to Karolinska from Harvard. So we're exploring now how we may take advantage of this technology to mass produce human cells in vivo and subsequently use them for transplantation. So these are some of the last slides. I have to acknowledge, thank the league of extraordinary collaborators that we have had the luck to work with in the past many years. I told you about Roger Hajar, but the immediate past president of the American Heart Association, Gordon Tomaselli, immediate past director of biomedical engineering at Johns Hopkins, both of them are from Johns Hopkins. We've been working together for years, and they are coming to Hong Kong next week to explore ways of continuing to work together, not only in cardiovascular diseases, but also other disciplines of reparative medicine. So Chris Chan from West Institute, he was also here in Hong Kong a couple of weeks ago for the Keystone Symposium. And of course, Kevin Costa and Michelle Kine from the US, and also all the other trainees. So this is the list of trainees from the program. Many of them have graduated and moved on to new ventures already, and the whole project couldn't have been completed without their support. And these are the faculty investigators involved in the project. Heartfelt thanks to all of them who have contributed. Some have retired, some have left Hong Kong, some have moved on from one institution to join another. Some may not even work together again in future, but many have become very close colleagues and friends and sticking together, moving on to new directions or ventures, collaborative programs. So to conclude, did we make some good progresses? Yes, we're not shy to say that we have. Did we make mistakes? Lots of them, and way even more. And even up to today, I still apologize to the folks, to Paul Tam, to Paul and John about the mishaps that we have encountered. There were there were things that have been said that we wish that we could reverse, and things that have been done and we wish that they could be undone. But the fact is that rest assured that we have learned a lot from in the process and moved on. And indeed, we never promised that we're not going to make mistakes or even fail. But we promise that we will never fail to try it. And once again, thanks to RGC, we're glad that this chapter is now closed. But I'm sure that long-range benefits will continue to come in the many years in future. Thank you very much.
Thank you, Professor Li. Please remain on stage. May we now invite Professor Lo again to host the Q&A section with Professor Li, please.
Yeah, thank you very much for Ronald to tell us his work on the cell-based heart regeneration. So any questions from the audience on his project? So as a layman, according to your project aims to develop the cell-based regeneration medicine to help the patient with the heart disease. So will you expect in the near future this technology, for example, the stem cell transplant can actually apply in the clinic?
Near future time window is a very relative term. I'm an optimist that I think in our lifetime we're going to see therapies and different ways to transplant cells into the heart. So I'm very confident that it will happen. But is it going to be 3 years, 5 years? I think it's very hard to predict. And it's easy for anyone to want to be the first to be doing it, but I think more importantly is to become the first to succeed. And that's required some planning. So Holy Grail obviously is to regenerate cells and put them into the heart to repair. But for now, before we get to that point, one step at a time is that before we can put these things, the cells, tissues, patches into the heart, human heart, we are now using them for facilitating drug discovery. And in the process, we're going to learn more and more about these constructs, and as such, we know more about them. That's a good thing because we know how to fine-tune them in future to improve the efficacy and also the safety before we put them into the humans. So the world, we can see that at about 1.5 years or 2 years ago, there were very few or virtually zero clinical trials. But now everything is just budding, and you're starting to see a lot about macular degeneration, spinal cord injury, diabetes, artificial pancreas, and also Parkinson's diseases. So heart is going to be one of them, but it's going to take a little time,
Is Professor Li. I want to ask is that the structures that is contained in the cylinder that is showed on your slide, is it a 3D model or 3D structure of cardiomyocytes together? That means your thumb is in the final weapons, the thumb-like structures. Is it made of a thin layer of cardiomyocytes?
Good question. A lot of people talk about 3D for heart tissues. But if you are talking about a tissue strip that we also have, and worldwide there are maybe about 5 or 6 groups that have the capability of making strips like this. But for me, it's still 2D, because it's not flat, it's not totally flat, but for me, it's still 2D because the motion is like this, right? So that's 2D. But for the human heart in the jar technology, it's really 3D. It's a pouch. It's a bag that is fully capable of ejecting pressure. So eject like the way that the left ventricle ejects blood. So we call it a genuine 3D structure. So as such, you're not measuring just the force, but you can measure the pressure. So at the same time, measure ECG signals and do the pressure volume loop. So our hope is that it's in a jar because it can be cultured this way, and you take it out of the incubator and you can make measurements like this and put it back into the incubator. So as such, it's really like the Langendorf heart preparation, except you're looking at human hearts, and they're sitting in a jar that can be removed from the incubator and put back, as you wish. It's about the size of your thumb. Obviously, it depends on how big your thumb is. So it sounds small, miniature, but if you compare that to like an adult mouse heart or just adult mouse heart left ventricle, it's actually not that small. But we strategically make it small. Why do we want to do that? So that we can mass produce, we can make more, improve the production, right? So it's not about mimicking the full heart or reproducing the full heart capacity or capability, but to mimic to such an extent that it can be an intermediate step between animals and human patients.
So you mentioned that one of the problem is those cardiac cells are immature. I wonder what are the consequences? So they cannot contract or the...
No, absolutely. So very good point. So immature in the sense that if anything, they are more like fetal or baby heart cells. And if you think about it, it might make sense because you're doing everything in culture. It's just like you get a baby, you have to allow some time for them to grow. And we know from developmental cardiology that the heart doesn't really grow until the baby is born, when the pressure and everything, is changed, right? So immature, it doesn't mean that they're not functional. It just means that they're not fully like adult, hearts. But in many ways, good enough is good enough if we were just using them to model. If they're good enough to make some predictions, it doesn't mean that they're not useful. And quite interestingly, as we put together these cells, immature cells, into different forms, including like a heart in a jar, a pouch, a bag, so somehow just the three-dimensional environment of configuration itself is able to drive maturation to a very large extent, as indicated by the functional properties and also the genetic properties as we do single-cell sequencing and all those to look at the genetic profiles. Yeah. But they are not like fully grown adult hearts yet.
So another issue you mentioned is the heart is also small size too. I wonder if it's possible to use an artificial scaffold to increase the size.
Yeah, we can try to increase the size. There are different ways of doing this for scientific curiosity and also for practicality. But as I said before, it's been strategically designed to be small because if you think about it like the size of a mainframe computer about 30, 40 years ago, as big as this room, what you want to do is not to make it bigger. You want to shrink it down to make it as small as you can, yet you want to be able to maintain the key features, functionality, and capability. And that's why we try to make it small for drug discovery so that we can make more. So think about if we have 100, 200 mini hearts sitting in an incubator, that's like having a population of genetically identical, or you can actually make them different hearts or patients sitting in an incubator, and you can repeatedly challenge and test with different pharmacological conditions. So the idea is to increase the throughput, accuracy, and sensitivity.
So for this mini heart drug screening in cell, whether you will generate this mini heart from different population, for example, just Professor just say that there's a lot of the mutation or the variation from the Asian, from the Caucasian. So maybe the drug response are different from based on the population. So whether okay.
That's a very great point, and the ability to personalize is extremely powerful. Yeah. So that means that we can make miniature hearts for each and everyone in the audience. And the idea is that, as we all know, traditional drug therapies have been designed largely based on Caucasian populations. But now, think about we can make miniature hearts for Northern Chinese, Southern Chinese, Caucasian, Jewish, and you name it, the different ethnicities, and look at the genetic differences without having to worry about what exactly the precise underlying genetic bases are. Because simple-mindedly, we just cared about the phenotypes. We just cared about how they respond differently. So that's a very powerful approach. And also, while we can make normal human hearts for screening toxicity, we can also make sick hearts that carry particular disease mutations such as dilated cardiomyopathy, genetic arrhythmias, and screen for efficacy. So run the different compounds in and see what can potentially improve cardiac function. That's actually exactly as for the spin-out company. So the first client happens to be a major global pharmaceutical company that wants to design a particular heart for a disease of interest so that they can particularly look at that population of patients.
Okay, thank you. Thank you. Any questions from audience?
Thank you, Professor Li and Professor Lo. The morning session of the symposium is coming to an end. Let us give a big round of applause to thank our facilitator and our presenters for their enlightening sharing. We will now have a lunch break until 2:30 p.m. Please come back on time and see you in the afternoon.